Crispr by Isabella Morales. CRISPR technology can be utilized in the editing of genomes. This particular technology can often be used to prevent diseases, genetically altering crops, and correcting genetic defects. It can be used to treat or correct Down syndrome, sickle cell anemia, and other similar genetic defects. CRISPR, short for CRISPR-Cas9, stands for clusters of regularly interspaced short palindromic repeats. CRISPR was developed based on the interactions between bacteria and viruses. Bacterial populations were often killed off by the effects of viruses. But in the situation that the bacteria did survive, the bacteria would take a section of the virus's DNA and save it in a sort of DNA archive called CRISPR. If the viruses were to attack again, the bacterium produces an RNA copy of that section of the bacterial DNA. This RNA is then fed into protein Cas9. Cas9 then scans the bacterium and compares all DNA that passes through it to the virus. Once it finds viral DNA, the protein can cut out the viral DNA, rendering it useless. Regarding the CRISPR technology, Cas9 can be programmed to cut out a specific section of DNA. What makes CRISPR so special is its ability to work with live cells, to control gene expression and study DNA sequences. This technology can be utilized in all sorts of cells, including human, animal, plant, and bacterial cells. Although CRISPR is an extremely powerful tool, it does have its limitations. CRISPR is very difficult to administer in large numbers to mature cells. Since mature cells have already multiplied, it is difficult to correct all the cells. Additionally, CRISPR does have the risk of editing the wrong gene. This can have severe and possibly deadly consequences. Although CRISPR can be used for many things such as immunotherapy, genetic disease treatment, drug development, and crop improvement, there are a lot of cons. For example, germlining, bioweapons, reduced genetic diversity, and a safety risk. Ethically, CRISPR technology does have some issues. CRISPR can be utilized to edit gametes and embryos. This can be utilized to fix any genetic disease or undesirable traits. But when this type of power is given, where does it stop? It is very difficult to restrict what can be controlled by CRISPR. This technology can begin to be used for enhancement purposes as opposed to therapeutic purposes. Many fear this technology will only be available to the rich, and additionally, there could be many objections to embryo manipulations and testing as a result of religion. CRISPR is still being developed and is relatively new technology. As time goes on, this technology will continue to progress. With the power of gene editing, researchers will have the ability to do things we have never dreamed of before. In the future, CRISPR could be utilized on mature cells and change things we never thought were possible, making the dream of eternal life a lot more achievable. Although in its early stages, this technology could radically change the way we live today. From designer babies to superhumans, the possibilities are endless. But with everything, we must be very careful with how this technology is used. Regulations and laws should be implemented as this technology continues to expand.